technology. And okay, we started with some not yet okay, we started with some history actually about traditionally how we modify the plants as we want, because the plants are very important food, but that is not fast enough, not good enough, not specific enough, so we also apply biotechnology or modifying the plants, which we call as food like crops or other type of food. Okay, so of course you have to know about how to do so, and we start with breeding the plants. So you have to know how we can breed an individual plant from just a single cell. So for plant, this is actually not that hard, because the plant cell are totipotent. That means if you take the correct parts of the plant, they can just go into a whole new plant from a single cell at the very beginning. But we still have option. We can have different ways of growing the plants, and you have to know about how to grow them, the basic component in the medium. You have to make sure everything needed for the plant growth are there, so that they have all the nutrients, all the chemicals, all the growth regulator, like something like hormones to stimulate their growth, so that they can grow from a single cell into an individual new plant. Okay, and we have choices. We can do colorless culture, we can do suspension culture. So you have to know about the difference. What are they? Why they are different? What is the difference after you culture it in this way, and what is the next step difference? How you can obtain the plants afterwards, or will there be any differences in the follow-up work on these two type of culture? But these are both possible. We can use solid medium or liquid medium depending on what you want. Okay, and some more detail during the culture. You have to make sure there is no contamination in the plant culture. So actually the same, no matter you're culturing the bacteria or plant cell or animal cell later, you always have to make sure there is no bacteria contamination, because you're growing some cells. If they are contaminated by bacteria or fungi, then of course they may not win in the competition. They compete for the nutrients in your culture medium, they compete for space. Even what is worse, they can be killed by the bacteria if they produce like toxin or some other thing. So you have to make sure when you're doing culturing, no matter plants or animal culturing, you have to make sure that it's sterile, because you don't want the contamination. And also you have to know how to keep it sterile. Okay, you have to do it by yourself in the lab, so you should be able to tell the importance and why. Okay, and then still some more technique when we are culturing the plant cell, how to stimulate their growth, how to keep it for longer time later, something like that. And then there is something called forward genetics and reverse genetics. So just some idea on you deduct from the function to the gene, or from the gene to the function. There is just some idea like that. And then we talk a lot about some features we can add to the plants if we do about recombinant technology on them. So basically what you want, okay? So for all production of transgenic organism is the same. So first of all, identify what you want. Okay, for example, I want the plant to be more resistant to herbicide. Then identify the gene that can give you this function. So identify the target gene, and then be able to take out the target gene by DNA isolation, and then be able to put the target organism, like put it into a crop, and then be able to screen to find out which plants are really transgenic now, because it is not 100% successful way if you try to put DNA in a new in another organism. So you also have to be able to screen. And if you go through all these steps, identify the gene, taking out the gene, put it into another organism, screening for the transgenic animal or plant, then you are able to get a transgenic organism, and you can get the feature you want. Okay, so for plants, some of these features are like having resistance to herbicide, so that you can spray the herbicide to your farms and killing only the weeds but not your crops. And we can also give them, so these are some detail of how we can do it in real life. And we can also give them a higher insect resistance so that they can fight against the insect by themselves. You don't have to add too much chemical, too much insecticide to kill the insect, because that will also like damage other wildlife or make them more resistant. So we want to make the plant defending for themselves by giving them insect resistant gene. Okay, and then this is what I briefly mentioned, producing these transgenic plants. So you have to identify the characteristic you want, identify the gene that gives you the characteristic, take it out, put it into another organism. So go through the whole step, the factor thing, the transformation thing, the screening thing, blah, blah, blah. And then you can grow a new plant if the DNA are incorporated in there successfully. Okay, so for plant, there are some specific thing we can, some specific technique we can do in transformation, because there are bacteria that naturally infect the plant cell, so we can make use of them. Okay, so this is plant specific because they infect plants, so we can make use of them. So this include the T plasmid and V plasmid. So they're so special that they can be used in plants only, or they can be used in plant very well. So make sure you know about them, know about their story. So we have T plasmid. We talk about how they work. So they break a piece of them, they get inside the plant cell, and then they are stick with the plant DNA. So make sure you know about T plasmid, V plasmid, because we can use them for plant cell transformation. Okay, so after this, if they are successful, or you don't know if they're successful or not, then you have to do detection. So this is still about how to put the DNA into the plant cell. Okay, so apart from T plasmid, of course there are other way. Okay, so may not be the same as bacteria transformation. So make sure you know about some specific way for doing bacteria or plants or animal. So for plant cell, we have T plasmid, V plasmid, very specific, but we also have something like the fold dip, particle bombardment, gene gun, something like that. So make sure you can name a few. Okay, and of course we also always have CRISPR gene editing. Okay, it is like the final tools. You can always use this on all type of cells. Very specific, very fast, very high throughput. So, but we are not focusing in here. We will talk about this later again. But make sure you know what is a CRISPR, mainly the CRISPR-Cas9 system. Okay, and then so no matter which method you use to produce a transgenic plant or try to produce a transgenic plant, you have to go through screening to detect which one is really transgenic, so that you can further culture that plant to produce transgenic offspring. Okay, so make sure you know how to detect for plants. We can have some gene added together with your target DNA, like producing some antibiotic resistance, or allow them to light up, to show up themselves, some reporter genes. So some example like fluorescent, blue light. So make sure you can, again, name a few examples here. Green fluorescent, blue light, something like that, for you to detect the transgenic plant cell. Or you can also use PCR, sequence the whole thing to find whether the new gene are inserted or not. So there are always many options. And then there is about some, again, very practical issue. We make the transgenic plant. We want to make transgenic plant, and then they are GM food. But people are concerned about the new DNA being added into the food. So we want to minimize the extra gene in there. So we have to be able to remove those reporter genes, selectable marker, because they are there for screening only. So after you make sure the gene is the plant is transgenic, you don't need them anymore, because they are not the gene that give you the new feature in the plant. So we have to have some way to take them out again, so that people will be a bit more acceptable towards the GM food. So the tools, we have the cryptose system. We talked about this in quiz already, so you should be able to tell how they work already. Okay, so don't go through it again here. And then the later works, so still about breeding, testing. So there's a many extensive follow-up work actually. After you produce a transgenic animal, you still have to test it, screen it to see whether the new feature are really stable, whether the next generation, the next generation, the next generation still have the new feature bringing by the new gene. Before you can make sure they are safe enough, their features are stable enough so that they can become a new type of GM food for putting into the market. Okay, so some example of GM food already available in the world. Okay, so we have many presentations on GM food last week. So I think you are very familiar with this topic already. And lastly is how to detect the GM food. The one way, very straightforward way is still PCR, checking their gene, checking the DNA, the length, the size. If we know about like the fragment or the piece of DNA that can be the new gene, then we can use primer to copy the specific area, check for the fragment. We can further have some M time to cut them up. Okay, so basically just PCR and then gel electrophoresis with some reference band and then check against it to tell whether they're GM or not. Okay.